Okay, let's unpack this. Our mission today is to cut straight through the dense molecular forest of cancer research and really isolate the central engine that drives a malignancy, and that's the oncoprotein. We're going to focus pretty intensely on one of the foundational, the defining capabilities that cancer cells acquire. It's a concept known as self-sufficiency in growth signals.
That's a great place to start. And if you think about it this way, normal cells, they need an external invitation to start dividing. They need a growth factor.
Right.
This deep dive into the source material shows us exactly how cancer cells not only write their own invitations, but also how they manage to cement the accelerator pedal to the floor permanently.
Bypassing everything.
Everything. All the external regulatory mechanisms, they go right out the window.
And that's really the hook, isn't it? At the heart of this whole process, the source material reminds us, is this fundamental event, non-lethal genetic damage.
Right. Carcinogenesis, the origin of cancer, it's not one big catastrophic failure. Yep. It's the result of complementary incremental mutations that accumulate over time. It's a stepwise process.
A slow-motion genetic siege.
That's a good way to put it. And the handful of mutations that actually give the cell a selective growth advantage, the ones that contribute to this malignant phenotype, those are what we call driver mutations.
Okay.
So the cell just keeps accruing these little changes until it finally flips a switch and acquires these terrifying capabilities, these hallmarks.
So the key insight here is really the accumulation, the stepwise nature of the failure. It's not a single lightning strike.
Absolutely not. And when we look at which genes are primarily targeted by these driver mutations, they fall pretty neatly into 4 functional classes.
Okay, what are they?
So first, you have the growth-promoting proto-oncogenes. These are the normal, responsible genes that, eventually become these hyperactive problems.
The good guys turn bad.
Exactly. Second, the growth-inhibiting tumor suppressor genes, which are our cellular brakes and governors. And third, you have the genes that regulate programmed cell death or apoptosis.
And then there's a 4th class. It's often overlooked, but it seems profoundly important in just accelerating the whole process.
It's the key accelerator, you're right. The 4th class involves the genes responsible for DNA repair.
The editors.
The editors themselves. If you imagine a cell's genetic code being constantly checked for errors, the DNA repair genes are doing that checking. When damage happens to these repair genes, especially early in the process, the editor essentially quits.
And then chaos ensues.
Total chaos. Every subsequent change, every typo in the genetic code goes uncorrected. This leads to what we call a mutator phenotype, and this guarantees that all the future driver mutations accumulate at a catastrophic rate. It just fuels the rapid evolution of the cancer.
That concept, the mutator phenotype, that feels crucial. It really explains the aggressive speed you see in advanced cancers. It's a self-perpetuating system of genetic failure.
It is. But for our focus today, we're squarely on that first class, the proto-oncogenes, and how they evolve into oncogenes.
Okay, so for you listening, what is the crucial distinction here? Because it sounds like there are 2 sides of the same genetic coin.
They are, but that distinction is the core of our dive. A proto-oncogene is a normal, regulated cellular gene. Its product promotes growth, yes, but only under very specific, tightly controlled conditions.
Like wound healing or something.
Exactly, during wound healing or development. An oncogene, however, is the mutated or overexpressed version of that gene.
And here's the key analytical point, right? These oncogenic changes, whether they're a point mutation, a gene amplification, or a translocation, they result in a gain of function.
That's the phrase, a gain of function. And that's why we call them dominant in genetic terms. The activated oncogene product just overpowers the normal gene product from the other chromosome.
So it's driving this autonomous growth factor-independent proliferation as a permanent unilateral override switch.
It is. So the mutation doesn't just reduce function, it actually creates a dangerous new function.
And that's what makes them such powerful disease drivers.
Exactly. And to really understand the scale of what the cancer cell is doing, we first have to review the normal growth process. It's hijacking.
Okay, the relay race.
The relay race. Physiologically, growth signal transduction is very orderly. You have a growth factor that binds to a specific receptor on the cell surface. This kicks off a cascade of cytoplasmic signal transducing proteins. The signal travels inward to the nucleus, where it activates transcription factors that, turn on genes, leading ultimately to cell division and growth.
And what we are about to detail is how cancer achieves this self-sufficiency by hijacking or bypassing or just permanently activating nearly every single step in that cascade, starting with the growth factor itself.
Right. So let's start at the very top, the membrane. The very interface where the cell normally has to wait for that external invitation to grow.
Okay, so let's unpack this. How does a cancer cell successfully bypass the need for an external growth factor that, say, a neighboring cell would normally provide?
One of the simplest and, frankly, one of the most brazen ways is to establish what we call an autocrine loop.
Autocrine loop.
Yeah. Normally, growth factors are made by one cell type and they act on a neighboring cell of a different type. That's called paracrine action.
Right.
But some tumors become masters of self-stimulation. They actually start producing the exact growth factors that they themselves are responsive to. It creates this continuous internal stimulus loop.
So they're literally writing their own invitations and then binding them to their own receptors, just constantly yelling "go" to themselves.
Precisely. A very clear illustrative example is found in malignant brain tumors like glioblastomas. They often express both the platelet-derived growth factor, or PDGF, and the corresponding PDGF receptor at the same time.
So it's a closed circuit.
It's a perfect closed circuit. Or you see sarcomas overexpressing, transforming growth factor, TGF-α, and its receptor. The critical insight here is that the tumor has completely eliminated its reliance on the surrounding tissue for that necessary go signal.
Okay, so moving from the factor to the receptor itself, the source material really emphasizes the absolute importance of receptor tyrosine kinases, or RTKs. These are the antennas on the cell surface.
They are. RTKs are these massive transmembrane proteins. They span the entire cell membrane. They have an external binding domain and this powerful intracellular enzyme domain, the tyrosine kinase domain.
And the normal system has very strict rules for these.
Very strict. A growth factor binds outside, the receptor activates transiently, it dimerizes, it autophosphorylates, and it kicks off the internal signaling. And crucially, it then quickly shuts off.
But the oncogenic versions are different.
They're designed for continuous output. They are constitutively active, always on, constantly delivering these mitogenic signals regardless of what's happening outside the cell.
So how does the cell achieve this permanent on state in such a complex protein? What are the mechanisms?
We see three primary genetic mechanisms.
Yeah.
And the diversity of the approach is really what makes cancer so challenging to deal with. First, you have point mutations.
Okay.
A well-known example is the ERBB1 gene, which codes for the epidermal growth factor receptor, or EGFR.
Right, in lung cancer.
In the subset of lung adenocarcinomas, yes, single-letter typos in the genetic code, often within that intracellular domain, can cause this profound conformational shift in the receptor protein. It permanently mimics the activated state and frees its tyrosine kinase domain from any regulation.
A single amino acid change, and the accelerator's just stuck down forever.
That's the effect.
Yeah.
The second mechanism is gene amplification. So if you look at ERBB2, which codes for the HER2 receptor.
HER2 in breast cancer.
Exactly. In about 20% of invasive breast carcinomas, the ERBB2 gene is massively amplified. This means the cell ends up with hundreds of copies of the gene.
So you just get a massive overexpression of the HER2 receptor protein on the cell surface. A huge overexpression, so even if the individual receptors are behaving normally, activating and deactivating, the sheer overwhelming quantity of them results in this continuous, powerful signal transmission.
It's like a signaling traffic jam that never clears.
That's a perfect analogy. And finally, the third mechanism we see is gene rearrangement. This is where parts of two different genes get fused together because of a chromosomal deletion or a translocation, and they create a new chimeric protein.
Like a Frankenstein protein.
Basically. The source mentions the EML4-ALK fusion gene found in a subset of non-small cell lung adenocarcinomas.
That specific example is fascinating. A deletion on chromosome 5 causes parts of the ALK gene and the EML4 gene to fuse, correct?
Correct. And the resulting EML4-ALK chimeric protein is specifically designed for perpetual growth. The EML4 part has a domain that promotes self-association, which forces the ALK tyrosine kinase domain to dimerize and persistently activate, even without a ligand.
So you have these three different mechanisms, point mutation, amplification, rearrangement, and they all achieve the same result, that constitutive tyrosine kinase activity. What does that diversity of mechanism tell us about how we should approach therapy?
It tells us two critical things. First, that cancer is opportunistic. It just takes the path of least genetic resistance in different tissues. And second, the therapeutic approach has to be tailored exactly to the mechanism. For an amplified receptor like HER2, the successful drug is often a monoclonal antibody like Herceptin that physically blocks the massive quantity of external receptors.
Right.
But for a point mutation or a rearrangement like ALK or a mutant EGFR, you usually need a small molecule kinase inhibitor that targets the specific perpetually active enzyme inside the cell.
So that molecular diversity is why personalized oncology is so critical.
It's everything. It's why we have to know the specific driver.
That makes perfect sense.
Yeah.
And we've seen how targeted drugs against amplified HER2 in breast cancer or against mutant EGFR and ALK fusions in lung cancers, they've just proven the importance of these molecular targets. So now let's follow the signal indoors. We've established the external signal is hijacked. The next crucial question is, what happens if the internal components, the ones meant to transmit and shut down that signal, are also permanently broken?
Now we move from the cell surface antenna to the internal wiring, and the undisputed champion of the short circuit here is the king of oncogenes, RAS.
RAS.
RAS proteins are these small membrane-associated G proteins that operate as the crucial internal switch for many of these RTK signals.
Could you explain that switch mechanism for us again? Because this seems absolutely fundamental.
It is. Normally, RAS cycles between an active signal-transmitting state when it's bound to the high-energy molecule GTP and a quiescent inactive state when it's bound to GDP.
On and on.
On and off. The crucial off switch is provided by a class of enzymes called GTPase activating proteins, or GBAS. JPs don't just turn RAS off. They accelerate RAS's own intrinsic ability to hydrolyze GTP back to GDP by over 1,000-fold. GPs are the necessary, highly efficient brakes on RAS activity.
And the mutational problem here is that this brake fails.
Completely fails. In cancer, point mutations in the KRAS, HRAS, or NRAS genes abolish that critical GTPase activity, the mechanism that lets it convert GTP back to GDP.
So the protein gets stuck.
The mutated RAS protein becomes trapped in the perpetual active GTP-bound form, sending continuous pro-growth signals down the line. It is a profound, irreversible failure of the off switch.
And the prevalence of this failure is just staggering. This makes RAS arguably the most important single oncogene discovery ever.
It's the ultimate therapeutic bottleneck. RAS mutations are implicated in 15 to 20% of all human tumors globally. But the statistics truly skyrocket in certain highly aggressive, hard-to-treat cancers.
Like pancreatic cancer.
About 90% of pancreatic adenocarcinomas have a RAS mutation, usually KRAS. You also see it in 50% of colon cancers and 30% of lung adenocarcinomas. Targeting RAS effectively has been one of the holy grails of oncology, precisely because it's so widespread and so critical to these intractable diseases.
You know, the insight here is the sheer redundancy of the cancer mechanism. If you don't have an activating RAS mutation, you might have just lost a protein that normally holds RAS in check.
Right.
Which brings us back to the tumor suppressor genes, even within this section on oncogenes.
That's a perfect illustration of how complementary these systems are. Think about the tumor suppressor gene NF1. NF1 codes for a protein called neurofibromin 1, which is a key GP, that molecular brake we just talked about.
So losing NF1 does the same thing as turning RAS on.
It produces the exact same end result as an activating RAS mutation, uncontrolled RAS activity, because the mechanism to turn it off is just gone. Cancer either presses the accelerator by mutating RAS, or it cuts the brake line by losing NF1.
So once RAS is perpetually active, the signal doesn't just stop there. What are the specific, powerful signaling arms that it engages next?
RAS activation stimulates two major downstream arms, the MAPK cascade and the PI3K-AK pathway. And components near the top of these cascades are frequent targets for oncogenic mutations themselves.
Okay.
For instance, BRF, a member of the RAF family of seronephranine protein kinases, is critical to the MAPK arm, which regulates cell division.
And BRAF mutations are infamous in melanoma.
In about 60% of melanomas, yes, plus a huge percentage of thyroid cancers. The activating point mutation in BRAF5V600E mutation constantly stimulates downstream kinases and transcription factors, completely overriding the need for RAS or RTK input.
And that's another case of oncogene addiction.
A classic one. The fact that targeted inhibitors against mutant BRF were so successful in melanoma was a major clinical triumph, again demonstrating that concept.
Then we had the other major branch, the PI3K-CHAP pathway. You mentioned earlier that this pathway sort of orchestrates the cell's entire construction program. What makes this pathway so special, beyond just driving proliferation?
The PI3-CACAP pathway is like the cell's general contractor. It's a master regulator for rapid growth and survival, not just simple cell division.
Right, because to create two daughter cells, a cell has to literally double its physical mass.
It has to double everything: new lipids, proteins, organelles. This pathway, which acts through the crucial mTOR kinase, is vital for anabolic metabolism. It stimulates glucose uptake, promotes lipid biosynthesis, and coordinates the entire protein synthesis process.
So the cell doesn't just want to replicate its DNA. It needs the raw materials, the physical bulk, to actually perform the division.
Exactly. And gain-of-function mutations in the PI3K family are extremely common. We see them in about 30% of breast carcinomas.
And what about the flip side?
Conversely, its crucial negative regulator, the tumor suppressor, PTN, is often lost in endometrial and prostate carcinoma. This means this pro-growth anti-apoptotic signaling is just completely unrestrained. The pathway gets attacked from both sides simultaneously.
That sets the stage beautifully for understanding another common way the cell creates a perpetually active signal, this time relying purely on a genetic accident, the creation of a new fused enzyme. Let's talk about non-receptor tyrosine kinases.
This brings us to a really canonical example of targeted oncology and really the genesis of modern cancer therapy: chronic myeloid leukemia, or CML, which is driven by the Philadelphia chromosome.
The Philadelphia chromosome.
It's a specific, well-defined chromosomal translocation, specifically T922.
Could you detail that translocation for us? Because it's such a profound molecular event.
It is. So on chromosome 9, you have the ABL gene. On chromosome 22, you have the BCR gene. The translocation fuses these two genes together to create the BCR-ABL chimeric protein. And this happened in a single, non-lethal accident.
And what is it about this new fusion protein that makes it such a powerful driver of cancer?
Well, the ABL component is a non-receptor tyrosine kinase. The resulting BCR-ABL protein has constitutive or continuous tyrosine kinase activity because the BCR part contains sequences that promote the self-association and spontaneous dimerization of the whole chimeric protein.
So it forces itself to be active.
This forced dimerization is enough to unleash the full, inherent, unregulated tyrosine kinase activity of ABL. It continuously drives growth-promoting signaling pathways without needing any external growth factor.
It's hard to overstate the initial shock in the scientific community when targeted inhibitors for this protein first showed that we could halt a cancer driven by a single translocation. It basically defined the concept of oncogene addiction.
Absolutely. CML cells, despite potentially having other mutations, they become totally dependent on this one oncoprotein BCR-ABL for their proliferation and survival.
It's like the central pillar holding everything up.
We think of it as the central lodgepole holding up an entire tent structure of oncogenic signaling. If you remove that lodgepole with a drug like imatinib or Gleevec, the entire structure collapses, and that leads to dramatic tumor regression.
That level of dependency is what makes the initial clinical response so revolutionary. But the source notes that this targeted therapy isn't necessarily a cure, right? Even though the response is incredible, why does the treatment have to be continued indefinitely?
It's because of the persistence of these rare CML stem cells. The vast bulk of the tumor cells, the ones that are rapidly proliferating, they are addicted to that BCR-ABL signal for survival and growth.
Right.
But there's a small population of primitive stem-like malignant cells that harbor the BCR-ABL fusion gene, but they seem to enter a quiescent or dormant state.
But they don't need the signal.
They don't seem to require the BCR-ABL signal for their basic survival, so the targeted therapy doesn't touch them. If you stop treatment, these cells slowly awaken, they spawn rapidly proliferating offspring, and the leukemia inevitably returns.
That introduces the critical complexity of cancer stem cells being resistant to this kind of targeting.
It's a huge problem.
A profound caveat to the success of targeted therapy.
Okay, so we've traced the signal from the membrane with the RTKs through the cytoplasm with RAS and PI3K. Now, all roads lead to the nucleus, where the decision to transcribe growth-promoting genes is actually made. And the master orchestrator that translates this constant go signal into action is the transcription factor, MYC.
MYC is the ultimate decision maker. It's what's called an immediate early response gene, which means it's normally rapidly and transiently induced by external signals like RAS-MAPK. Its concentration is tightly regulated and it's very short-lived.
But in cancer, that control is catastrophically lost.
It is. It turns MYC into the cell's general contractor.
Why do you call it the general contractor? What are MYC's broad activities that make it so powerful?
Well, it acts as the switchboard for all the growth machinery because cell division requires doubling the cellular mass, not just copying the DNA.
Okay, so what does it do? First, yes, it activates key components like D-cyclins, which are crucial for getting the cell cycle to progress into S phase. Second, it enhances ribosome assembly and RNA processing, which is essential for rapid and massive protein synthesis. Without new protein, you can't build two cells.
And third, it drives that critical metabolic change we hear so much about. It links the nuclear activity directly to resource management.
Absolutely. MYC upregulates a gene expression program that drives metabolic reprogramming. It activates glycolytic enzymes and glutaminase, which feeds the Warburg effect.
So it's getting the building blocks ready.
It's preparing the cell to build macromolecules like DNA, lipids, and proteins by ensuring a massive influx of carbon building blocks. It is literally making sure the cell has all the necessary supplies for its massive ongoing construction project.
Wow.
And finally, MYC contributes to the cell's immortality, basically, by promoting telomerase expression and regulating stemness.
So given its profound and widespread effect, how is MYC typically deregulated in cancer?
The classic example is the translocation seen in Burkitt lymphoma, T814. This moves the MYC gene, which is on chromosome 8, right next to the powerful gene regulatory elements, the enhancers of the immunoglobulin heavy chain, or IgH gene on chromosome 14.
And IgH enhancers are incredibly powerful, especially in B cells.
They are. In B cells, these enhancers are constitutively, highly active. They're driving massive protein production all the time.
So placing MYC under their control.
It causes massive, continuous overexpression of the MYC protein in those B lymphocytes. The cell is constantly using a high-octane B cell expression system to power its general growth.
And that explains the translocation mechanism. What about amplification?
Alternatively, we see gene amplification. This is common in neuroblastoma, where the NMYC gene is amplified, and in small cell lung cancer, where LMYC is amplified.
And what does that look like?
These amplified genes can manifest microscopically as these little extra chromosomal structures called double minutes, or as chromosomally integrated homogeneous staining regions, HSRs. But regardless of the mechanism, translocation, amplification, or just massive upregulation by upstream signals like RAS or WNT, the net effect is the same. Inappropriate, continuous, and high levels of this master transcription factor.
Okay. So we've covered the accelerator pedal, the oncogenes from the membrane all the way to the nucleus. But for growth to be truly unchecked, the speed limit has to be removed entirely. And that brings us to our next hallmark. Insensitivity to growth inhibition, disabling the tumor suppressor genes, or, well, cutting the brakes.
This is the necessary complement to oncogene activation. Uncontrolled growth absolutely requires both activating the accelerator and inactivating the brakes.
You need both.
You have to have both. Tumor suppressor proteins normally apply those brakes. They govern cell cycle checkpoints and respond to internal stress signals by shutting down proliferation. We really need to look at the two most important tumor suppressor genes, RB and P53.
Let's start with the governor of proliferation, RB, the retinoblastoma protein. What is RB's primary job in a normal, quiet cell?
RB is the critical negative regulator of the G1S cell cycle transition. That's the point of no return, where the cell decides whether or not to commit to division.
Okay.
In quiescent or resting cells, RB exists in an active hypophosphorylated state. And in this state, it acts like a molecular manacle. It binds tightly to and inhibits the E2F transcription factors.
And E2F is the transcription factor that activates all the genes needed for DNA replication. So by binding E2F, RB just arrests the cell.
Exactly. Holds it in check. But when a normal cell gets a growth signal, that signal leads to the activation of D-cyclins and their partners, CDK4 and CDK6. These kinases then hyperphosphorylate RB,
which inactivates it.
It inactivates RB, forcing it to release E2F. The E2F is now free to go activate the transcription of S-phase genes, and the cell is committed to division.
So how do cancer cells attack this really elegant system?
They achieve RB inactivation in three main ways. 1, by direct loss-of-function mutation in the RB gene itself. Classic examples are retinoblastoma and small cell lung cancer.
Okay.
2, more commonly, they acquire gain-of-function mutations that upregulate the proteins that inactivate RB, specifically by amplifying CDK4.
And the third way?
They lose the key inhibitor of the CDK cyclin complexes, which is another tumor suppressor called P16.
So the disruption of the RB pathway is functionally central to malignant transformation, even if the RB gene itself isn't the thing that's mutated in a specific cancer.
That is the ultimate paradigm of redundancy failure. At least one of the 4 key regulators of this G1S checkpoint RB CDK4 cyclin D, or P16, is dysregulated in the vast majority of human cancers.
The system has safety checks, but cancer finds a way to disrupt it, usually at the weakest upstream point.
Exactly.
Okay, now to the most famous tumor suppressor of all, P53, often called the guardian of the genome. It's mutated in over 50% of all human cancers. Why is it so central?
Loss-of-function mutations in TP53 are truly ubiquitous. The p53 protein is the central monitor of cellular stress. It gets activated by multiple profound inputs: massive DNA damage, shortened telomeres, hypoxia, and crucially, oncogenic stress.
That's oncogenic stress.
That's the stress caused by excessive, inappropriate pro-growth signaling from things like mutant RAS or overactive MYC.
So P53 is the quality control officer that gets activated when the cell starts acting out of line. But how does the cell keep P53 in check normally?
Normally, P53 is kept at very low levels. It's rapidly degraded by a protein called MDM2. MDM2 is the natural killer of P53.
Okay.
When stress occurs, P53 levels have to rise dramatically. This is achieved in a few ways. For example, when oncogenic signaling elevates the expression of the P14ARF protein, that P14ARF protein specifically inhibits MDM2.
So by stopping the killer, you allow P53 levels to build up and do its job.
That's it. It's fascinating that activating an oncogene like RAS or MYC itself stresses the cell so much that it triggers this complex molecular alarm bell.
And once it's activated, P53 can thwart this neoplastic transformation by initiating one of 3 terminal outcomes.
The first and most common is transient cell cycle arrest. P53 induces the transcription of a CDK inhibitor called P21. P21 inhibits the CDK cyclin complexes, which keeps RB inactive and arrests the cell at that G1S checkpoint.
It's buying the cell breathing time to repair the damage.
Exactly. But if that repair fails, the cell has to choose a more terminal option.
Right.
So the second option is senescence. This is a permanent, irreversible cell cycle arrest where the cell can't divide again. You're essentially putting the damaged cell into permanent retirement.
And the third and most definitive protective mechanism
Is apoptosis, or programmed cell death. P53 directs the transcription of pro-apoptotic genes like BAX and PMA, which ultimately trigger the mitochondrial pathway of cell death.
So the critical insight here is that for a cancer cell to grow vigorously using activated oncogenes like RAS and MYC, it absolutely must first inactivate P53.
It has to. If P53 function is lost, DNA damage goes unrepaired, the cell bypasses the arrest, and driver mutations just accumulate rapidly, leading to malignant transformation.
And this has massive clinical relevance.
Huge. Tumors with wild-type or normal P53 respond much better to conventional genotoxic therapies like radiation and many chemotherapies because those treatments rely on P53 to induce apoptosis in the damaged cells. A non-functional P53 confers profound drug resistance.
Wow. Okay, before we leave the breaks, we need to cover 2 other major tumor suppressors involved in key signaling pathways, APC and PTTN.
Right. APC, adenomatous polyposis coli, is famously the gatekeeper of WNT signaling, particularly in colorectal cancer.
And WNT signaling promotes growth.
It does. A major function of the APC protein is to form this specific destruction complex that identifies and degrades a molecule called beta-catenin, keeping it out of the nucleus and inhibiting the WNT growth pathway.
So beta-catenin is the messenger of WNT growth, and APC is the molecular chaperone that just throws the messenger into the trash bin.
That's a very accurate way to put it. And loss of APC function, which happens in 70 to 80% of sporadic colorectal carcinomas, disrupts this destruction complex.
So beta-catenin builds up.
Beta-catenin accumulates in the cytoplasm and just floods the nucleus, where it partners with transcription factors like TCF. This constantly stimulates growth genes, including our old friend MYC and cyclin D1.
So the cell behaves as if it's under constant maximum WNT stimulation, driving this relentless proliferation.
Correct.
And then we have PTEN, which regulates the flow of the PI3-CACT signaling cascade we discussed earlier.
PTEN is a lipid phosphatase that's encoded by a gene on chromosome 10. It acts as the negative regulator, essentially the brake on the PI3KT cascade. PTEN's job is to reverse the action of PI3K.
And when PTEN is lost?
When PTEN is lost or mutated, which is a very frequent event in endometrial, breast, and prostate carcinoma, the PI3KT pathway is continuously active. This promotes continuous growth, synthesis, and anti-apoptotic signals.
It reinforces the idea we started with. These pathways are often attacked by complementary mutations. An oncogene activates the pathway, or a tumor suppressor removes the brake.
Exactly. A PI3K gain of function or a PTEN loss of function, same outcome.
This molecular understanding of the accelerator-on break-off system hasn't just been an academic exercise. It has led directly to some revolutionary treatment strategies. Let's look at the advancements and new frontiers in growth control that this knowledge has unlocked.
Indeed. The ability to identify these hyperactive mutant kinases, BCR, ABL, mutated ERBB2, mutant BRAF, has resulted in highly specific drugs, the kinase inhibitors, which were once pure science fiction.
And these drugs can halt cancer in its tracks if the cancer is truly addicted to that single signaling molecule.
If it's addicted, yes.
We saw the immediate dramatic success in CML, but the challenge remains resistance. The source material makes it very clear that these targeted therapies aren't usually curative, especially in advanced solid tumors.
That is the single most daunting clinical problem: tumor heterogeneity and evolution under pressure. The tumor isn't a static entity. It's a dynamic, evolving population.
Right.
When subclones within the tumor are exposed to an effective inhibitor, those that survive often possess acquired secondary mutations that allow them to just sidestep the drug's effect entirely.
Can you give us an example of how a cancer finds a bypass like that?
Absolutely. Take lung cancers driven by a mutant EGFR. A drug initially works wonders, but resistance eventually emerges. One resistance mechanism involves a secondary point mutation within the EGFR gene itself, like the T790M mutation, that physically prevents the inhibitor molecule from binding to the enzyme's active site.
So the cell keeps its addiction, but the drug just can't plug the hole anymore.
Correct. Or, even more sophisticated, the cancer cell might activate an entirely different parallel signaling system, what we call bypass signaling.
A whole new route.
A whole new route. For example, the tumor might amplify a completely different receptor tyrosine kinase gene, like MET. By massively overexpressing MET, the cell reroutes the growth signal around the blocked EGFR receptor, maintaining its self-sufficiency through a new, unchecked channel. This ability to adapt is why combination therapies are increasingly necessary.
If the first generation of research focused entirely on cell division signals, the next frontier, driven by ththose PI3, KKT, and MYC findings, seems focused on the energy and the material requirements for growth, the metabolism itself. Let's spend some time detailing the Warburg effect.
The Warburg effect, or aerobic glycolysis, has puzzled researchers since it was first described. The question is, why do cancer cells, even when oxygen is plentiful, rely overwhelmingly on the seemingly inefficient glycolytic pathway?
Right. It only generates 2 molecules of ATP per glucose.
Instead of the highly efficient oxidative phosphorylation in the mitochondria, which yields 36 ATP, it seems totally counterintuitive.
For a rapidly growing entity to choose inefficiency in energy production, yeah.
And that's the insight that new research provided. It's not about maximizing ATP yield, it's about maximizing raw material production. Oxidative phosphorylation converts glucose entirely into CO2 and H2O. That's great for energy, but they're metabolically inert. A rapidly dividing cell needs to double all its components, DNA, RNA, proteins, lipids, for 2 complete daughter cells. And that requires vast amounts of precursor molecules.
So glycolysis is preferred because it produces the right kind of intermediates that can be immediately siphoned off for building things.
Precisely. Aerobic glycolysis provides the necessary carbon moieties, the metabolic building blocks. For instance, intermediates from glycolysis can feed the pentose phosphate pathway, generating ribose 5-phosphate, which is essential for nucleotide and DNA synthesis. And crucially, NADPH, which is vital for reductive biosynthesis, like making lipids and protecting the cell from oxidative stress.
So the cell is choosing structural integrity and construction readiness over pure energetic efficiency. It's constantly in a state of construction.
Absolutely. And this detailed molecular understanding of metabolism led to a really surprising and profound discovery. Oncometabolism driven by mutations in core metabolic enzymes, specifically IDH.
This is truly a frontier, focusing on a non-oncogene that suddenly acts like an oncogene.
It is a profound change in perspective. Mutations in isocitrate dehydrogenase, or IDH, which is a key Krebs cycle enzyme, occur in gliomas and acute myeloid leukemias.
And these mutations don't just stop the enzyme from working.
They give it a new, perverse enzymatic activity. This mutated IDH begins producing massive, unnatural amounts of a molecule called 2-hydroxyglutarate, or 2-HG.
And 2-HG is the oncometabolite. What does this aberrant molecule do inside the cell?
2-HG acts as a powerful competitive inhibitor of several epigenetic regulatory enzymes that normally require a different metabolite, alpha-ketoglutarate, as a cofactor.
Yep.
Crucially, this includes enzymes of the TIT family, which are vital for maintaining normal DNA methylation patterns. By inhibiting these epigenetic enzymes, 2-HG causes widespread abnormal patterns of DNA methylation and histone modification. It essentially reprograms the cell's entire gene expression profile.
So a faulty metabolic enzyme acts upstream to create an aberrant metabolite, which then acts as an epigenetic poison, silencing or misexpressing growth and differentiation genes. That links metabolism directly to genetic regulation and transformation.
And this mechanism led directly to drug development. Because a mutated IDH protein has a structurally altered active site, scientists were able to design specific small molecule inhibitors to target the mutant IDH.
Which halts 2-HG production.
Exactly. It halts 2-HG production and can even reverse some of the epigenetic changes, providing new therapeutic avenues for these specific leukemias and brain tumors.
That's incredible. Moving from controlling the energy flow to controlling the cell's very identity, let's look at the fascinating realm of differentiation therapy. This sounds like trying to force a cancer cell to grow up and get a job instead of proliferating wildly.
That is exactly the goal, forcing the malignant cells to exit the immature proliferative state and mature into short-lived functional cells. The classic and most successful example is an acute promyelocytic leukemia, or APML.
Right.
APML involves a T1517 translocation, creating the PML-RRA fusion gene.
And why does that specific fusion cause leukemia?
The fusion protein acts as a dominant negative. It blocks the normal myeloid progenitor cells from differentiating into mature neutrophils. They just get stuck in a rapidly proliferating immature blast state. The core problem is the blocked differentiation pathway, not just the overproliferation itself.
So the counter strategy involves using a drug that targets the fusion protein and just forces differentiation.
Yes. The strategy relies on high doses of all-trans retinoic acid, or ATRA. The normal retinoic acid receptor alpha binds retinoids, like ATRA, and activates genes needed for maturation.
But the malignant fusion protein, PML-RRA, is a potent repressor of those genes.
It is, at normal physiological retinoid concentrations, but high pharmacologic doses of ATRA can overcome this repression.
Ow.
Pharmacologic doses of ATRA bind to that PML-RRA complex and cause a necessary conformational change that displaces the repressor complexes. This unlocks the transcription factor and forces the malignant progenitors to complete their differentiation process.
So they become neutrophils.
They turn into short-lived, mature neutrophils, which naturally die off, clearing the leukemia without the need for traditional cytotoxic chemotherapy.
That is a total reversal of the malignant phenotype. It's truly revolutionary because you're not killing the cell, you're simply telling it to grow up and die naturally.
It's a beautiful example of molecularly targeted therapy.
Finally, before we wrap up, we should briefly touch upon non-coding RNAs and their role in regulating all this growth machinery.
It's another critical layer of regulation. The best characterized non-coding RNAs are microRNAs, or miRs. These are small RNAs that inhibit the translation of messenger RNAs. They act as gene regulators, fine-tuning protein output. In cancer, altered miR expression can either promote or suppress tumor development, adding another layer of complexity to this self-sufficiency hallmark.
So we have the bad ones and the good ones in the miR world.
Right. Oncomers are those that are overexpressed and repress tumor suppressor genes, or genes that promote differentiation. Conversely, the loss of certain tumor-suppressing miRs contributes significantly to cancer.
Is there a good example of that?
A key example is in chronic lymphocytic leukemia, or CLL. Deletions affecting the miR15 and miR16 cluster are very common. The loss of these miRs leads to the massive, unchecked upregulation of the anti-apoptotic protein BCL2.
Which promotes tumor cell survival.
Exactly. This shows how controlling cell death and controlling growth are intimately linked, even at the level of these tiny regulatory RNAs.
This entire journey, from the growth factor receptor all the way down to the metabolic pathways and the tiny microRNAs, it really paints a comprehensive picture of relentless molecular self-optimization by the cancer cell. Let's bring this full circle now.
We have dissected how cancer achieves its most fundamental hallmark, that self-sufficiency in growth signals. We started at the cell membrane, looking at receptors like EGFR and HER2, which get stuck in the on position by mutations or amplification or rearrangement.
Right.
We moved into the cytoplasm with perpetual signaling from transducers like RAS and PI3K, which don't just accelerate division, but also orchestrate this massive cellular construction program.
And we ended in the nucleus with the master orchestrator, MYC, which translates this continuous signal into mass production of cellular components and fuels that metabolic shift.
Right.
The key insight is that the cancer cell has flipped the accelerator switch permanently, and simultaneously, it has cut the brakes by disabling key tumor suppressors like RB and P53. This continuous, relentless drive for proliferation and growth is what allows the disease to bypass the body's natural regulatory framework and become so aggressive.
And this molecular complexity, the necessity for complementary stepwise mutations in oncogenes and tumor suppressors and metabolic regulators to acquire all of the hallmarks of cancer, is the key takeaway. Cancer is a failure of multiple interlocking regulatory systems. It allows the cell to bypass growth control, evade death, and acquire immortality. Understanding the molecular basis of the accelerator and the brake is the foundation of modern oncology.
It is the ultimate system failure, but it's also the blueprint for successful targeted intervention. And that brings us to our final provocative thought for you to consider. Targeted therapies today, like kinase inhibitors, they primarily attack the consequences of these mutations, the perpetually active protein, with the aim of killing the cell or halting its division.
But given our growing and profound understanding of oncometabolism, like with IDH and the Warburg effect, and the tremendous clinical potential of differentiation therapy, like with ATML and ATRA, we have to ask a crucial paradigm-shifting question.
What if the future of cancer treatment isn't focused on killing immortal cells, but on finding a reliable way to reverse the cancer's stem-like state? What if we could engineer a systemic method to force these seemingly immortal self-sufficient cells back into a normal, finite differentiation pathway, rendering them completely harmless?
It's a question of whether we must destroy the enemy or if we can simply find the molecular switch that forces it to put down its weapons and integrate back into normal, finite tissue function.
That's something to mull over until our next deep dive.